Good day, this is Dr. T, and welcome to the final video in my metabolomic tour of Wikipedia. So let's end on one that is very not Homo sapien centric, photosynthesis. So, of course, this is absolutely critical for us because if you want to eat something, at some point, photosynthesis basically had to take place. So let's talk about this. So obviously you remember from way back when photosynthesis, you've got your plant, it takes light, takes carbon dioxide, takes some water, makes oxygen, and it makes sugar for the plant. So hey, we could do this particular reaction over here to make ourselves some nice sugar. By the way, the fact that photosynthesis is making what starts out basically as glucose is one of the reasons why plants do so much with carbohydrates. They are carbon-rich, nitrogen-poor, so one of the limiting nutrients they have is nitrogen. That's why the importance of nitrogen fixation, Haber-Bosch, and whatnot. But they've got plenty of carbon. They're pulling it from the atmosphere. They got more than they ever want, so they're going to build everything out with the highest percentage of carbon that they can. So they're going to go for, hey, carbohydrate, good old polysaccharides, whenever they can do it. Hence the use of cellulose and then lignin, which is, well, not a polysaccharide. It's definitely a carbon-oxygen containing polymer. Okay, so let's look at photosynthesis. Photosynthesis is broken into kind of two major blocks. You've got the light reactions. These are going to take water, turn it into oxygen. The oxygen is waste. Their plants aren't going to use it for the photosynthesis. Now, one of the downsides of photosynthesis, it requires light. And as you know from solar cells, or just looking outside, you only get the power for half the day. The other half of the day, it's dark. You know, moonlight is not going to be strong enough to get any meaningful photosynthesis going. So you're going to have to store that. And then that's where the Calvin cycle, otherwise known as the dark reactions, take place. In this case, you're going to take carbon dioxide, you're going to take the energy you get from the light reactions, and you're going to turn that into sugar. And by doing that, you can then burn the sugar doing good old respiration like we animals do, or you can use it to build stuff. So yay. Well, let's look at these guys. Just kind of a point of reference. Photosynthesis is going to be taking place in the chloroplast. Chloroplast has thylakoid membranes. These are structurally a similar idea to a mitochondria, but obviously completely different. Okay, so we can look at our photosystems kind of in two ways. Let's look at the pictures in order they appear. So obviously this looks a lot like the electron transport chain because it's an electron transport chain. It is, however, in this case for the chloroplast. So a couple of major features. One, we are doing proton gradient. This is actually kind of inverted from the direction that the mitochondria do. So for the innermost part of it, that's where the hydrogens are being pulled from in the mitochondria, and this is where they're going to in the chloroplast. I mean, it's a minor detail. It's the basic same idea. And we're going to do this proton gradient, and we're going to use that to power some ATP synthase. It's very similar, you know, we're doing the exact same thing. Now, you will notice that whereas the mitochondrial electron transport chain started out with NAD and ended with water, we're going to start with water and we're going to end with NADPH. Remember, the P is a phosphate group attached to the bottom of it, otherwise it's the same molecule. And that allows the cell to have two accounts, essentially, so that you've got the NADH account, which is reduced electrons or high energy electrons that you can use for energy production. And you have the NADP and NADPH, which is high energy electrons that you're going to use for anabolism, for building things up. And so this is actually the first time we're talking about an anabolic pathway, where you are building things up. Now, we can look at this a little bit differently. Let's make this bigger. There we go, we're bigger. Okay, so you start out, you've got the oxygen evolving complex. That's going to pull electrons from water. Once again, it's basically the exact opposite of what complex 4 was doing. You're pulling electrons from water, you're making the oxygen in the process. Oxygen, once again, is a byproduct for this guy. This goes into your photosystem too, but This was discovered first, then we discovered this. Now, what's not shown on this one, and I kind of wish it was, is these photosystems. These are, kind of your chlorophyll is at the center, which is doing the, the function. You got your chlorophyll and then the proteins around it, but you also have secondary pigments. And these are going to be things like beta carotene, which are able to absorb light, but at slightly different frequencies. And then by doing so, it gives more photons the ability to transition that energy into one of your two photosystems. And so, boom, we get the electron goes in there. We add a photon that gives us energy. That makes it a higher energy electron. We take some of that energy out. That becomes ATP. Go to photosystem 2. That kicks it up. And then we eventually turn NADPH. Okay, so once we've done this, we've got energy. So now we're going to take the energy and we're going to use this energy. And this is called the dark reaction just because it doesn't require light, but it doesn't need to be in the dark, and typically it's not. So this is the Calvin cycle. I always get this one confused. This is the Calvin cycle. This, interesting enough, uses rubisco. This is arguably the most common enzyme on the planet. There's a lot of plants. And what's going to do is it's going to take the energy from the light reactions, and it's going to use that energy as well as the high energy electrons from the NADPH that were previously made in the light reactions. And it's going to take a carbon dioxide, and it's going to eventually add it into, and after several steps, it's going to be able to produce the precursors for gluconeogenesis. So we're going to be able to take the carbon dioxide, go through a cycle, and we're going to, and notice there's a lot of steps on this thing. And then we're going to take this, and this is one of the precursors for gluconeogenesis. So yay, gluconeogenesis. Now, this is what we call a C3 pathway. And most plants, especially a lot of the plants that we eat, are going to be doing this. Now, there are modifications to this pathway. So some plants will do what's called a C4 pathway. So in this case, it's still got the Calvin cycle. Everything we just talked about still takes place, but they add a little bit of extra complexity. So this can be between two different cells. Alternatively, it can be the same cell, but it's separated temporarily. But you've got a separation. And what we're doing is we're taking the carbon dioxide, we're pulling it out, and we're sticking it onto something. And then we're using that to store it. And I would say something, because it's almost always a 4-carbon molecule that you produce, but you've got a little bit of a different style. There's several different ways of doing this. So we're going to store it, and we either shuffle it to a different cell where the carbon dioxide can be released and then go into Calvin cycle, or we will just store it and let it sit there, and then later we'll do the Calvin cycle. And the reason we're doing this is that it turns out rubisco, remember that first part of the Calvin cycle, needs a fairly high concentration of carbon dioxide. If it doesn't, it can have problems, and it can start using energy for nothing. And it can also cause some damage to the cell. So what we want to do is keep the carbon dioxide high. Now, you're like, okay, so, well, the way you keep carbon dioxide concentration high is you open the stoma. You let more air into the leaf. Well, when you have the stoma open, more air comes in and more water goes out, which, if you're in a nice wet environment, yeah, that's fine. Alternatively, what you can do is you can slow things down, use a little bit wasteful for the energy. You get some of the energy back, but you still have to shuttle everything around. It's an absolute pain. But you can open the stoma on one, let these grab the carbon dioxide where it can handle a lower concentration, open them up, then shut the stoma. Now you take the carbon dioxide out, you shuffle it over, and you do this. This way you can open and close your stoma, and thus you don't lose as much water. For CAM plants, which use a somewhat different process but similar, they use, the whole thing takes place in one cell. They'll open their stoma at night, let the carbon dioxide in, store up as much of it as possible, close the stoma as the day goes, and then do the light reactions in the Calvin cycle during the day. These would be usually your succulents. The downside, of course, is that you can't open your stoma during the day because it's dry. You're in a desert. You're a succulent. The whole point is to run on next to no water. So they run out usually by about 10:30 in the morning. They are out of stored CO2, and thus photosynthesis just kind of idles. They have all the energy they need. They just can't get carbon dioxide because, they don't want to open their stoma. This is why cactuses grow very, very slowly. They are really limited. So yeah, that is photosynthesis. Just kind of scrolling down. Oh, sweet. Okay. So over here we have a spectra of our chlorophyll, and we have two different chlorophyll. These are the wavelengths that they absorb well. So, obviously it's not the entire spectrum. Down the middle, this is where green is. This is why plants are green. Chlorophyll does not absorb green, whereas other pigments, whereas now since it doesn't absorb it, it gets reflected, hence plants are green. The secondary pigments are going to absorb, kind of in this area and in this area, and they're able to absorb the photon, transfer the energy of the photon to the chlorophyll. And thus you get that energy, you get a much broader wavelength. And now, of course, for us, the secondary pigments can be useful, like beta carotene. And this is why, even though we think of beta carotene as carrots, it's also rich in a lot of your dark green veggies, because the darkness of that is coming from the fact that it's absorbing a lot of energy, or in the a lot of the spectrum. Let's see if I can butcher that anymore. Okay. With that said, have a wonderful day.